This lecture is an introduction to omics. In this lecture, we will gain insights into the basic concepts of structural and functional genomics, as well as get introduced to proteomics and transcriptomics. The World Health Organization defines genetics as the study of heredity, while genomics is defined as the study of genes and their functions and related techniques. The two sciences differ in their approach to the study of the gene and its functions. The main difference between genomics and genetics is that genetics scrutinizes the functioning and composition of the single gene, whereas genomics addresses all the genes and their interrelationships in order to identify their combined influence on the growth and development of the organism. Before we proceed further, it would be a good idea to recapitulate what we already know about the genes and the DNA. Deoxyribonucleic acid, or DNA, is a chemical compound that contains the instructions needed to develop and direct the activities of nearly all living organisms. The DNA molecules are made up of two twisted pairs of strands, often referred to as the double helix. Each DNA strand is made up of four chemical units called the nucleotide bases. These comprise the genetic alphabets A, T, G, and C. Bases on opposite strands pair specifically. An A always pairs with T, while a C always pairs with G. The order of the A's, T's, C's, and G's determines the meaning of the information encoded in that part of the DNA molecule, just as the order of letter determines the meaning of a word. This constitutes the DNA sequence, the code. An organism's complete set of DNA is called its genome. Virtually every single cell in the body contains a complete copy of the approximately 3 billion DNA base pairs, or letters, that make up the human genome. So literally, each and every cell of the human body is capable of providing the complete information that is required for constructing the individual or the organism. With its four-letter language, DNA contains the information needed to build the entire human body. A gene traditionally refers to the physical and functional unit of heredity. It is a sequence of nucleotides in DNA that codes instructions for making a specific protein or set of proteins. Located on 23 pairs of chromosomes packed into the nucleus of a human cell, genes direct the production of proteins with the assistance of enzymes and messenger molecules. Specifically, an enzyme copies the information in genes' DNA into a molecule called messenger ribonucleic acid, or the mRNA. The mRNA travels out of the nucleus and into the cell's cytoplasm, where the mRNA is read by a tiny molecular machine called a ribosome, and the information is used to link together small molecules called amino acids in the right order to form a specific protein. This process is known as translation. Proteins make up the body structures like organs and tissues, as well as control chemical reactions and carry signals between cells, and play a plethora of other functions essential for the survival of the cell. So if a cell's DNA is mutated or damaged, an abnormal protein may be produced, which can disrupt the body's or the cell's usual function and may lead to diseases such as cancer and apoptosis. Now, with this brief recapitulation of what we know about genes, genetics, and basic cellular processes, let us now delve into the process or the technique of omics. Omics technologies are also referred to as high-dimensional biology and encompass the cells, tissues, and organisms in a method that integrates the data from various platforms. Omics detects the genes, this branch is known as genomics. Omics that detects the mRNA, studies the mRNA, is known as transcriptomics. Likewise, proteins are detected in proteomics, while metabolomics focuses on metabolites. The integration and interrelationship between networks of biological processes is termed as systems biology. We'll talk more about omics in detail and system biology later in this lecture. Applied majorly for accurate understanding of normal physiological processes and gaining knowledge related to disease processes, which involve screening, diagnosis, and prognosis that provides an understanding of the etiology of disease. So omics are basically utilized for the holistic understanding of the various processes occurring in the body. In biology, the suffix omics refers to huge biological molecules. The broad analysis of large biological molecules was needed to be studied in detail as conclusion of Human Genome Project in 2001. Here, this figure summarizes the cascade of omics developments. So with the knowledge of the DNA sequences and genes through Human Genome Project, it became evident that genetics only cannot provide answers to most of the questions of human biology. This led to the development of Transcriptomics, Proteomics, and Metabolomics. Genomics is cognitive, comparative, functional, metagenomic, neuromics, personal genomics, epigenomics. So these are the various branches of genomics that are in existence today. Transcriptomics studies the mRNA that has been formed in the entire cell, that is transcriptomics. Proteomics may be further categorized into immunoproteomics, nutriproteomics, proteogenomics, and structural genomics, while metabolomics is categorized all into metabolomics and metabolomics. We'll talk more about it in later occasion. Focusing on genomics, it is a study of a set of genes. Refers to the interdisciplinary study based on evaluating the structure and function and mapping of the genomes. This term was coined by Tom Roderick in 1986 on mapping the human gene. The possible and highly researched areas under genomics include the functional genomics, metagenomics, and epigenomics. Structural genomics. Structural genomics is aimed at determining the structure of all protein fold families encoded by genes of living organisms. It basically focuses on the study of the protein products of genes. It provides insights into molecular architecture and mechanistic properties of the protein kinases, protein phosphatases, and phosphoprotein interactions and their domains. Structural genomics determines the structure of almost all the proteins in a cell or organism. The three-dimensional structures are important for the functional analysis of proteins in a cell and for rational drug design. While functional genomics is a branch that integrates molecular biology and cell biology studies. It deals with the whole structure, function, and regulation of a gene in contrast to the gene-by-gene approach of classical molecular biology technique. Here we understand the gene-gene interactions and how the overall genes in an organism interact to bring about and sustain life. It aims to relate the phenotype and genotype on genome level and includes processes such as transcription, translation, protein-protein interactions, and epigenetic regulation. Functional genomics involves comprehensive analysis to understand genes, their functional roles, and variable levels of protein expressions in an individual. Functional genomics refers to the development and application of global. Here, the global means genome-wide. Functional genomics refers to the development and application of global experimental approaches to assess gene function by making use of the information and reagents provided by structural genomics. It is characterized by large-scale experimental methodologies combined with statistical or computational analysis of the results. Functional genomics uses genomic data to study gene and protein expression and function on a global scale, or the genome-wide scale, or the system-wide scale. Here the system refers to the organism or the cell which is under study. So functional genomics uses genomic data to study gene and protein expression and function on a global scale, focusing on gene transcription, translation, and protein-protein interactions. So the functional genomics terms is used to describe the assembly of data to better understand the form and function of the cell. So much is already known about the mechanisms that govern the interaction of cells with its environment, and it is this base of knowledge that will act as a primer for solving puzzles of biology through functional and structural genomics. DNA, RNA, and proteins act together to maintain the integrity of the cellular environment, and how they interact with each other is the information accessible through many newly developed high-throughput technologies. By applying high-throughput technologies to the information already known about how cells respond to the environment, which is a part of functional genomics, the development of efficacious drugs with few side effects through intelligent design will be possible. So this is how the understanding of functional and system genomics, structural genomics, can help us to design better drugs. So functional genomics is a wide approach for predicting functions and interactions of genes and their products. An explosion of gene sequence information has posed a major challenge of identifying genes and determining their function. Today we very easily can get the sequence of DNA through high throughput technologies, get a DNA molecule sequence, identify the genes, but question arises, what's the function of that gene and how its activity is or expression regulated by the interaction with other genes. Functional genomic advances have led to the development of high throughput techniques that enable expression profiling of specific cell types. These exciting technologies, when combined with solid experimental design and validation using alternative molecular and cell-based strategies, comprise a paradigm that is useful for assessing mechanisms underlying the pathophysiology of many disorders. So through this knowledge of functional genomics, we have a holistic and overall view of the processes that are occurring at the gene level, how these processes are getting transferred or translated into protein-protein interactions, and how it leads to the maintenance of human health. And ultimately, this knowledge helps us to identify the pathophysiology or the physiology of the diseased individual. Transcriptomics encompasses the knowledge and information related to the mRNA. The presence of mRNA in the sample reflects the abundance level of the corresponding gene. A gene which is more highly expressed has more corresponding mRNA in the cell. So gene expression involves the detection and classification of mRNA mixture in a specific sample. The goal of gene expression profiling is to differentiate the mRNA mixtures from different samples. Contrary to genotyping, gene expression categorizes the level of gene expression. So here we are not talking about which gene is present, or we are not talking about the structural information of the gene, but we are now referring to the level of the mRNA in a cell. So the gene which is more expressive has more mRNA, corresponding mRNA in the cell, and vice versa. The variation of the transcriptome can be seen over time between cell types and change according to environmental conditions or under pathological conditions. On the contrary, proteomics refers to the total proteins expressed by a cell, tissue, or organism. The proteome is highly variable over time. That means the entire protein content in a cell is highly variable over time, shows specific variations, and alters depending on environmental and physiological conditions. So proteomics is defined as a study that provides insight into protein functions in biological systems. So protein, proteome, and proteomics. So protein is a molecule of amino acids that performs much of the life functions. Proteome is a set of all proteins in a cell, and proteomics is the study of protein structure and function. As defined by National Research Council Steering Committee, Proteomics represents the effort to establish the identities, quantities, structures, and biochemical and cellular functions of all proteins in an organism, organ, or organelle, and how these properties vary in space, time, or physiological state. Proteomics is a multidisciplinary approach or a multidisciplinary science. It involves protein chemistry, mass spectrometry, genomics, bioinformatics, computer science, and separation science, which we usually call the chromatography, and certain other techniques. Now from genome to proteome. Here we have a brief illustration to show the journey from gene to protein. The human genome is located in the nucleus of a cell. The genes, parts of the DNA, double helix, are functional units of the genome. They hold all information necessary to create the proteins required in a cell. So genes are located along the thread-like structures called the chromosomes. Protein synthesis is the transcription and translation of specific parts of DNA to form proteins. From DNA, we obtain the mRNA through the process of transcription. Then this mRNA is translated into a sequence of amino acids which we call as a polypeptide or the protein through a process known as translation. You will learn more about transcription and translation in other lectures. Here we have a representation of the central dogma. This is self-explanatory. I don't think this requires much explanation. Metabolomics. The small molecules referred to as metabolites constitute the metabolome. The interaction between genetic, environmental, lifestyle and other factors results in metabolic phenotypes. Two identical individuals still cannot be identical in their metabolome. What I mean to say is, all the factors like genetic, environmental, lifestyle affect the metabolome of an individual. Interaction of metabolome with other biological macromolecules in the cell results in metabolic pathways. The metabolic profiles of biological sample represent the metabolomics, which are changeable and time dependent and have a wide range of chemical structures. In this lecture, this is what we have learned about omics, genomics, transcriptomics, proteomics, and metabolomics. Here we have some more additional resources to help you in the process of understanding these concepts. Please go through them and please provide your feedback on the lecture in the comment section in Google Classroom.